Hello, I'm Douglas Slotta. I'm a staff scientist at NCBI, which is the National Center for Biotechnology Information. If you haven't heard of us, we do PubMed, and we're also the primary data archive for genomic data in the world. I'd like to thank the three speakers this morning for providing all the background. I don't need to teach anything other than what a genotype is. It's basically the difference between your genome and someone else's genome. So to give a size of the data, the big bar on the left is the raw sequence data, the BAM files that are the short reads that are assembled to make the genome. And then the other two ones are showing how we compress it and bring it down. And then there's my little data set at the end. It's only small in comparison. We expect it to grow. In fact, this data over to the side here, in 10 years or so, we expect it to disappear. There's no reason to keep around once our sequencing technology is fully trustworthy. We can, you get a genome, you just have to store that. You don't have to store the short reads anymore. So our current data set is based on a 1,000 genome project. There are approximately 40 million sites for mostly SNP data, but other short indels that we track variation currently with humans. Currently, the 1,000 genome project has 1,092 people, and they eventually plan to stop around 2,500 people. So the increasingly misnamed 1,000 genome project. It's not just the genomic data at the point, but also the quality scores and everything else that provides those data points, if you can do that math in your head and see it doesn't add up. And we don't want to just keep around the most current data set. We also keep track of all the old data sets and possible mappings to different reference frames, which is what the human genome map is. So I like to call this the accession.version.version, just to confuse people. Our data source is called VCF. It's a tab-based text format. Each row is a variation site, and each column is a data sample. And we want to access this in two orthogonal methods. One is by list all the variations, list all the genotypes for a given variation, and then list all the variations for a given sample. And it turns out to be a very large, sparse matrix. If only there was some way to handle that. We have a production browser in place. This is a mashup of a lot of resources at NCBI. We have our sequence viewer, we have our ideogram, and our Entree search for gene locations, but this is the part of it I care about, which is the genotypes. You can look at the population data in aggregate. Here each row is a population, or also a subject, and each column is the variation in its position. And you can see exactly what a person would have. This is based upon SciDB. You can see that how long it takes to load and to run our data, but once it's up and loaded, it's pretty fast. We can get about 13 genotypes a second, 13 genes a second, which doesn't sound too fast until you realize that's 1.13 million genotypes a second, and this is just based off a small cluster right now. Of course, I did all this work myself with the help and assistance of these people. Thank you very much.